Excellent. So I first want to thank the organizing committee for this invitation to speak today. I wish I'd be with you in, I guess, a nice weather in Vancouver, although it's becoming sunnier here over time. So I first want to disclose that I have no relation with industry. I'm a researcher funded by the CRC and the CIHR and the CFI. So first, we're just going to go back in the rationale for using CP or CCP. I guess most of you are well aware of the McGuire and Redden paper in 1918. Published during the Spanish flu. So the infusion of convalescent plasma is not new. And theoretically, it should neutralize a virus when given with antibodies against this virus and prevent further replication. This is all theoretical, as you know. There's been a few publications on this over time, and there's a systematic review published 7 years ago of 700 patients with SARS-CoV-1 infection or severe influenza, showing that CP was found to be associated with mortality. This was not only RCT data, though. So what's the rationale for using CCP in COVID-19? So the majority of patients who recover from COVID-19 illness develop antibodies, as you well know, 2 or 3 weeks after the infection. So these antibodies are easy to detect through ELISA or other quantitative assays. So that's kind of the rationale and the justification that brought several countries and especially the United States to start giving CCP during the pandemic when we had no treatment and no evidence-based treatment to give to our patients with a high mortality before vaccination, as you can easily imagine, widely used to treat patients, especially in the United States, with close to 100,000 patients who received CP during the pandemic. But this is all outside clinical trials. So there was a use of several interventions, treatments in China when it started, in Europe after, and in the States before we can really start putting some evidence out there. But when we started to think about what we should be studying, like convalescent plasma, the question was, will the COVID-19 pandemic be the same research failure as the H1N1 pandemic? For those who remember, at the time we had a fairly short pandemic, but there was several interventions that were thought to be beneficial that could never be studied because the wave was over once REB approval was obtained. So to prevent such thing to happen, the REMAP-CAP trial was developed. So it's an international multi-center clinical trial platform. So it's not only a trial, it's a clinical trial platform that allowed to go way beyond a simple trial looking at one intervention against no intervention or a placebo. And it's international with several countries being involved in this initiative. So it was planned before the COVID-19 pandemic exists. So it's a platform that is out there for which we had REB approval for community-acquired pneumonia, and it was some sort of sleeping for a potential pandemic to come. We thought it was to be an influenza pandemic, but it happened to be a SARS-CoV-2 pandemic. So REB approval was already there at several sites in several countries, ready to go for a pandemic to come, for a community-acquired pneumonia, viral or bacterial, and to study potential treatments that we ignore could be potentially seen as beneficial, but in a way that the platform was made to rapidly integrate new interventions. So it was activated at the end of February of 2020 with several potential treatments that were used in China or thought to be potentially beneficial, antivirals, lopinavir, ritonavir, hydroxychloroquine that you've heard about, steroids, immunomodulator, anticoagulants. and antibodies against SARS-CoV-2. So several domains of intervention to study different treatments that were used at the bedside without evidence. So how does it work? It's every patient with a suspected or proven COVID. At the time, we didn't have a PCR test. So we enrolled at the beginning of the trial suspected patients that were contact, because provinces and some countries couldn't cope with the amount of patients coming in, and enrolled into two strata of severity. Severe, so ICU-like a patient, ICU-like meaning that they need to receive ICU treatment regardless if they are in the ICU or outside the ICU, and moderate, so those on the ward on regular oxygen nasal prongs, as an example. So different domains of intervention. So a patient can be, could and still can be randomized in more than one domain of interventions. And each domain includes one or more therapy, one or more treatments, but they are only enrolled in one treatment or no treatment or another one within the same domain. So it's a platform that is designed with Bayesian analyses, with models, statistical models, making that there is no fixed sample size. It's all based on a posteriori probability of an effect, a posterior probability of an effect. So it's adaptive, meaning also that there is ways to stop an intervention when some criteria of futility are met, or to adapt the randomization when there are criteria of effectiveness that are met. And there's a possibility to add interventions down the road. So we're now studying other interventions and have modified some domains into REMAP-CAP to keep studying interventions that are potentially useful to treat COVID-19 patients that are admitted. What's our primary outcome? It's the number of days alive without the need for organ support at 21 days. So it's basically not being on the number of days not being on mechanical ventilation, vasopressors, or ECMO at 21 days. And if a patient died, the number of days this patient will be attributed will be -1. And if there's no day with organ support, it's going to be 22 days. So it's an ordinal scale out of 22 points. So over time, we have enrolled close to more than 11,000 patients in REMAP-CAP, and have randomized these patients into 20,000 interventions, including 2,000 in the immunoglobulin domain, which enrolled its first patient on May 5th, 2020. So we were pretty fast for a trial that involves an intervention that is much more complex than giving a pill of hydroxychloroquine every day. So these drug trial within REMAP-CAP started very early, about two months before. So it's pretty fast. Over 2 months, we were able to start enrolling in REMAP-CAP, and this is for sure with the help of our blood banking agencies, CBS and Quebec in Canada. So we published our results in JAMA a few months back. So our inclusion criteria in the pandemic were adult patients admitted to hospital with an acute COVID-19 infection, patients who for which death was imminent within 24 hours, or where there was a limitation of interventions to be discharged, or if more than 14 days have elapsed, were not eligible. So patients were randomized in a stratified way into a severe state, so those that are very sick on high flow oxygen or intubated or on non-invasive ventilation or on vasopressors, and those on the ward on regular oxygen or without oxygen. So stratified randomization, patients were to receive either 500 mL or 2 units of ABO-compatible convalescent plasma within 48 hours of randomization, or no convalescent plasma in the control group. So donors of plasma must have had a history of COVID-19 infection documented with a PCR at time of infection or positive serology. And the collection was done at least 14 days after the resolution of symptoms through an ELISA directed against the spike of the virus, the spike protein of the virus, or by a neutralization assay. So different ways. We didn't know at the time what was the best product to use. So some flexibility was given from country to country. So from an appearance point of view, as you know, CCP are no different than fresh frozen plasma, except that they contain detectable SARS-CoV-2 antibodies. Our primary outcome, as mentioned earlier, days alive and free of organ support at day 21. And secondary outcomes, all-cause mortality, 28-90 days, ICU hospital stay, classical outcomes that we will observe in ICU or in critically ill patients with thromboembolic events and SAA as outcomes. So as it is a Bayesian, or it was a Bayesian design, there was no predetermined sample size, but as opposed, predefined triggers for trial conclusion. So there's regular analyses of each domains triggered by enrollment into each domains, looking at whether superiority is obtained into the model. And superiority is very, very conservative, as you see. It's a posterior probability to have a proportional odds ratio greater than 1 at 99%. So 99% posterior probability of having a proportional odds ratio of greater than 1. So very, very conservative for superiority. When futility, it's a 95% posterior probability of having a proportional odds ratio lower than 1.2. So these were the two triggers for concluding the trial with a response adaptive randomization. So over time, we enrolled 2,000 patients, and in the two arms, most were enrolled in severe arm of the trial. Only 86 were enrolled in the moderate. In the United States, we had one site in Pittsburgh that randomized patient because they used convalescent plasma at the bedside. 11 patients were randomized to receive delayed plasma as opposed to plasma or no plasma. So about 1,000 patients per group were enrolled. So we observed no effect at 21 days of convalescent plasma based on our ordinal scale. On the right side, you can see with the color code, red being death and dark blue being having 21 day without support. So as you can see, there's no difference between the two groups for our primary outcome. When we look at subgroups, there's no difference for other secondary outcomes and also subgroups. However, so no difference whether they were hospitalized for more days than fewer than 72 hours or more than 7 days, if they were positive or negative PCR, or whether they were on mechanical ventilation. But the subgroup of patients with immunodeficiency, 126 patients, although were not statistically significant, showed a direction of effect that was clearly in the potential positive side. So no, not significant at all. So we haven't shown anything there, but it kind of raised some hypotheses thereafter. So a few months after, the recovery trial was published, showing similar results, more patients, but including ward patients, so including the moderate. So it's a mix of both, around the same number of patients in the severe state and a significant number of patients with moderate on the ward. Another trial that you're probably aware of, the Concord 1, published around the same time, showed also no effect of convalescent plasma overall on the ward for mortality or intubation at day 30. A systematic review randomized controlled trial. There's been a few ones since now showed no effect, mainly driven though by REMAP-CAP and recovery data because there's the larger trial in the literature. 2 outpatient trial, one published about 6 months ago, if I recall well, enrollment in ER, no effect on the composite of admission, need for ER or urgent care or death, 500 patients. But last week, as some of you are probably well aware of, another outpatient trial by our colleagues in the States showed that among 1,200 patients, there was a decrease in the hospital admission rate in the convalescent plasma group. This trial was conducted up to at the beginning of the Delta wave, so exclude patients in the Omicron wave. So what explain all these findings? Have we used suboptimal products, wrong population, timing of administration, or simply there's no effect regardless? It's a good question because when we look back at the potential subgroup of immunocompromised, it goes in the completely other direction than patients that were not immunocompromised. So that raised our thoughts that we and justify the relaunching of the trial in this new population of immunocompromised patients, critically and non-critically, with a new product. So we are restarting the trial, looking only at immunocompromised patients, a population, as you know, that is it doesn't respond very well to vaccination. We use a very pragmatic definition, the Apache 2 criteria of immunocompromission, which are already collected in REMAP-CAP. It's a very pragmatic trial. We're going to be randomizing patients on the ward and in the ICU. We'll use a new product, so high-titer plasma from convalescent vaccinated donors with a minimal requirement of adding 20,000 units per ml on the Roche Alexis anti-SARS spike assay, which is associated with a neutralizing antibody titer of 1 out of 320 against Omicron, based on our previous plasma that we collected in the UK in the previous trial. So that's the minimal requirement for enrolling in the trial. And these are data from our plasma in the UK showing that with a vaccine, 1 dose of vaccines, there's a really different response of neutralization against other variants. These are not Omicron for sure, but it shows that there really is a difference with these patients, as we know, with one vaccine that are convalescent. So we'll be using 2 units. Again, so around 500 mils within 48 hours of randomization, and we'll be stratifying randomization into moderate and severe, but also if patients receive monoclonal antibody, yes or no. So you'll say, okay, if they receive monoclonal antibody, are they going to already have antibody? So we know sotrovimab is probably not anymore useful in the for the AB.2 variant, the new Omicron variant. So we know that previous, as you know, monoclonal weren't useful or aren't useful at all on the Omicron. So these monoclonal antibody have a very short activity against new variants, and most of them aren't anymore useful pretty quickly. Same outcome measure. So days alive and free of organ support at 21 days, and same secondary outcome measures. So no predetermined sample size, but based on what we observed in our subgroup analysis, we estimate that we're going to be needing from 200 to 500 patients based on the models at different posterior probability. And the same predefined trigger for trial conclusion, superiority, defined as more than 99% posterior probability of an odds ratio of greater than 1. So it's a regular analysis, response adaptive. So in summary, there's no benefit observed in the first REMAP-CAP trial that was replicated also in recovery thereafter, and also in Conquer 1 on the ward. So in critically ill, there was no effect. But this is an unselected population, so there's a potential but unknown benefit in selected population of immunocompromised patients, and also using a more selected product of vaccinated convalescent donor. So the REMAP-CAP trial should be able to answer this question, and this such type of trial is a lot of work. There's my colleague, close colleague Srin and others, and John Marshall, that are helping lead this trial in Canada, and others around the world. It's funded by CIHR here, but we cannot ignore the huge amount of work by CBS, REMAP-Quebec in the first trial, and CBS in the next trial. That I want to thank for the work for the first trial, and also for the work to come, and all donors for sure that are without whom we couldn't do such type of study and put some evidence out there. Thank you very much.
I see Dr. Conway with his hand up already. Thank you, Alexis. That was great.
Alexis, that was fabulous. Bow to you and everybody else who does this kind of clinical work. That's why I don't do clinical work anymore. It's hugely challenging. For the future trial, I'm surprised you think you'll be able to do this with just 200, 300, or 500 patients, and particularly given this clinical situation that has changed so much over the past 6 months. And then and I'm not sure, how long do you think this trial will take to complete? And are you not concerned about changes in the clinical situation, whether due to the virus changing itself over the coming 6 months, year, or whatever it's going to take to draw conclusions on a very difficult project? I don't mean to pan it, but I'm really just concerned about how you're going to interpret the findings.
Oh, we all have the same concern. There is challenges here. That's for sure. One thing to remember: this is an international trial. So we have we enroll in Australia, New Zealand, mainland Europe, UK, States, and now in some sites in Asia, Japan. And in this particular trial within REMAP. So the scope of the trial is very large, so it's global. So in Canada, we're probably going to be representing perhaps 20% of that population. We believe it's going to take probably closer to 2 years or 18 months with all the sites we have. We have in total more than 300 sites in REMAP-CAP. But not all sites will be enrolling in the convalescent plasma trial. So that's one thing. With your second concern is another one that we have, and each time there's a new variant, we kind of think, okay, is it going to be working again? So that's why we looked at previous plasma and looked at the neutralizing effect of with the 20,000 unit per ml on the Roche Alexis assay. It provides a 1 out of 640 for Delta, 1 out of 320 for Omicron. We haven't done it for AB.2 yet, but we will. But we'll have to do it for sure for every new variants, and adapt because our minimal requirement is based on the Roche Alexis assay. So we'll have to see if it still fits a high titer in this with the new variant to come. But you're right. We cannot ignore that, and we have to act at each time there's a new one that becomes the main one infecting our patients.
Yeah, I'm sort of a bit baffled by all this for a host of reasons. Most complex clinical trial I've ever seen in my life, but good luck and keep at it. Is measuring neutralizing antibodies, neutralizing in an in vitro sense, is that the best measure of your immunity other than, say, something that looks, say, T-cell function? Because it strikes me you might be using a surrogate marker and basing your treatment on a surrogate marker. Does that make any sense to you or not? In other words,
I'm not an immunologist as you know, but this is a very good question, and that's the reason why in the first trial we accepted only as a minimal criteria, not necessarily going for a high titer, because we didn't know if neutralization was how it works. What we know now is it seems to be working better from subgroups as opposed to lower titer of neutralization, but it may be through the ADDC component, or there's. You're right that there is no perfect answer to that. It's I guess the decision here is. With the best we know, is it the best way to do it at this time, if we want to do it, or we just not do it and wait for the perfect answer? But this has been, as you can imagine, an ongoing discussion. And at the beginning of the trial, when we first started, there was strong opinionated leaders and team going for high titers, and others thinking, as you seem to be thinking, that it's not where the money is. So that's why the definition of the minimal requirement was more making sure that there is antibodies with a certain international level that can be compared from one country or jurisdiction to another.
Thank you. One other minor thing. You were very hard on yourselves with respect to superiority criteria. Was there a reason for that or not? So, I mean,
yeah, you're right. But that's Bayesian analyses. So we do a repeated measure. So in a frequentist approach, if we would be doing such type of repeated measures, let's say we use the more conservative one would be the Abetol-Peto criterion, which is 0.001. So it's the one that would have the lowest cost on the alpha at the end, but it's spreading your alpha value in a frequentist analysis. So at the end, the p-value, if we would be doing the same type of analysis that we're doing with this Bayesian approach, I guess our p-value of significance would be 0.001 at the end. So the Bayesian approach allows to do it, but we still need to have a very strict definition considering the repeated measures, interim analyses for continuing or not continuing the trial.
Okay, thanks. I've got it. It's an issue of multiple looks affecting the p-value. Okay, thanks a lot.
So, Alexis, we have one question from the ethers, which is saying, wouldn't it be easier and safer to use a drug like Paxlovid instead of using blood products, treating in the beginning of the infection or adding this drug?
It's a good question. It's another question, I guess. It's another research question. It could for sure be studied. As you know, Paxlovid is been studied in unvaccinated individuals. Whether we should be, we give it to immunocompromised individual. The NNT is 17 on variants that are before even before Delta. So does it work with vaccinated immunocompromised individuals? Do we have the same NNT? I don't know. It would be a good question. It's just another question to ask that hasn't been has been considered as being challenging because of the numerous contraindication and drug interactions with the use of Paxlovid, as you know.
Yeah, no, thank you. Anyone have any other additional questions? Okay, thank you very much, Alexis. That was really great.
Pleasure.